Is this on?
Yep, I think. Yep. Get going, please. Yes. Go for it.
Hello, everyone, and thank you very much for joining today's departmental seminar. Briefly, I'm Konstantinos Kaliviotis, a third-year PhD student and one of the PhD representatives at the Department of Bioengineering, working in the Pandazis lab. And I will be the host of today's seminar, which is the first of the seminar series selected by the PhD students of our department. So thank you very much once more, Naomi, for giving us this opportunity this year. Today, we have the pleasure to have with us Dr. Jennifer Lippincott-Schwartz, who is a senior group leader at the Howard Hughes Medical Institute Janelia Research Campus. So Jennifer Lippincott-Schwartz has an impressively diverse background, spanning from liberal arts to cell biology and biomedical research. She was a major in psychology and philosophy at the Swarthmore College, but after several years of teaching at schools, she entered the master's program in biology at Stanford University, working on DNA repair. Later on, she obtained her PhD in biochemistry from Johns Hopkins University in the mid-80s, while the start of the 90s found her being a staff fellow at the National Institute of Child Health and Human Development at NIH. Since then, she has pioneered the use of green fluorescent protein technology for quantitative analysis and modeling of intracellular protein traffic and organelle dynamics in live cells and embryos. Her innovative techniques to label, image, quantify, and model specific live cell protein populations and track their fate have produced vital tools used throughout the research community, while her own findings using these techniques have reshaped thinking about the biogenesis, function, targeting, and maintenance of various subcellular organelles and the macromolecular complexes and their crosstalk with regulators of the cell cycle, metabolism, aging, and cell fate determination. She is an elected member of the National Academy of Sciences, the National Academy of Medicine, the American Society of Arts and Sciences, and the European Molecular Biology Organization, and a fellow of the Biophysical Society, the Royal Microscopy Society, and the American Society of Cell Biology. She has a plethora of awards in her record, among them the E.B. Wilson Medal of the American Society of Cell Biology, the Newcomb Cleveland Prize of the American Association for the Advancement of Science, the Keith Porter Award of the American Society of Cell Biology, and the Theodore Linen Medal. In addition, Jennifer co-authored the textbook Cell Biology, and she was also president of the American Society of Cell Biology. Today, she will give a talk on emerging imaging technologies to study cell architecture, dynamics, and function. And with that, Jennifer, I think that we are ready to start. Thank you.
Thank you so much, Konstantinos. It's really a delight to be talking to everyone today. I'm sorry I can't be there in person. That makes a big difference in terms of my, being able to sort of understand, the excitement that's going on in your bioengineering department. But hopefully that will change, over the next year if everything goes well. I'm sort of located near Washington, D.C., and you probably heard in the news there's some real craziness going on there. So it's been quite a year, to say the least. But anyway, today what I want to talk about is the use of live cell imaging as well as electron microscopy approaches to characterize how cells are compartmentalized, eukaryotic cells in particular, and what that can tell us about the way cells function. So I want to start with this electron micrograph that was taken actually more than 25 years ago, which is just a thin slice through a eukaryotic cell, a fibroblast. And what you can see is that this cell is just loaded with subcellular compartments. But what's really interesting is that all eukaryotic cells are similarly subcompartmentalized into organelles that include the endoplasmic reticulum, the Golgi, endosomes, lysosomes, mitochondria, the nucleus, peroxisomes, and lipid droplets. And these 9 organelles really are a commonality of all eukaryotic cells. Even if you look at this small little Toxoplasma gondii, a parasite that can enter your intestinal tract, if you zoom in on the ultrastructure of that parasite, it is very parallel to what you see in a mouse fibroblast. So there's some basic fundamental rules in terms of the way that cells are engineered for their biochemical activities. As I mentioned, they're really key. There's a key 9 organelles. These organelles are organized as part of subcellular trafficking and organizational systems that serve very important functions. So the endoplasmic reticulum and the Golgi, for instance, are the organelles that are involved in secretory activity of cells. Endosomes and lysosomes are the organelles that are involved in uptake and degradation of materials from the outside world by the eukaryotic cell. And I should emphasize that bacteria and simple archaea cells, which are distinct from eukaryotes, lack these internal endomembrane structures. They don't secrete efficiently and they don't take up things efficiently. So this is a very unique characteristic of eukaryotic cells. The mitochondria is a very interesting subcompartment within eukaryotic cells. You all probably know it's key for energy production. The mitochondria generates ATP through its electron transport chain. But interestingly, people are now pretty convinced that it is an endosymbiont that was really a defining feature in the evolution of the whole eukaryotic kingdom. Basically, the thinking is that our archaea took up an alpha proteobacterium. That alpha proteobacterium that was inside that archaea ultimately became the mitochondria. And the interaction of that sort of alpha proteobacteria with the rest of the archaea host essentially led to this emergence of the eukaryotic system, which includes these internal membrane systems. The nucleus, as we move down this list here, is a unique feature of eukaryotes. It's where DNA, the genome, is being housed selectively and stored. But we also have these 2 other organelles, the peroxisome and lipid droplets, which are all about lipids, fat storage, detoxification. All eukaryotic cells have or have had these organelles associated with them. So they're really, we need to understand how, what these organelles are doing in greater detail if we're really going to understand how cells are functioning at a fundamental level. And to sort of emphasize, to underscore that point, we know that dysfunction in any of these organelles can lead to human disease. So what I'm showing here is just a list of medical conditions that emerge as a consequence of mutations in machinery that's associated with each of the organelles shown to the left. So for instance, many neurodegenerative diseases as well as bone disorders come from dysfunction in proteins that function within the endoplasmic reticulum or the Golgi or the secretory system. Niemann-Pick disease, Fabry disease are associated very uniquely with dysfunction in the endosome lysosome systems. Parkinson disease, diabetes, deafness, very much associated with mitochondrial defects, and so forth. So again, my goal actually as a biologist, as a cell biologist, has been to try to better understand what these organelles are doing within cells. And the journey has been really exciting and has been particularly benefited by the development of new imaging technologies, which have taken place over the last 25 years, starting with the real player in this world, I think, has been the development of the fluorescent protein, GFP. Back in 1995, the recognition that you can tag proteins with a GFP and have those proteins be expressed within any type of cell you want, and thereby enable you to visualize the protein populations and how they move within the cell, has really transformed our thinking about all of these intracellular structures, because we now had a way to tag these intracellular structures, and with the techniques that I'm going to be briefly highlighting in the course of this talk, shown to the left, we've been able to get really deep insights into the various characteristics of these organelles. So basically what my talk's going to be about is taking you through a very fast sort of movement through all of these different technologies that have been developed over the course of these 25 years that are sort of diving deeper and deeper into these different, well, it's enabling us to dive deeper and deeper into understanding various aspects of cellular organization. And I'm going to be talking about this from the framework of one particular organelle, which is an extremely important organelle, the endoplasmic reticulum, because it's not only the largest organelle in the cell, which spans from the nuclear, it forms the nuclear envelope, but it then extends out to the cell periphery as an elaborate tubular network. And ribosomes cover much of the ER's extensive membrane surface. These ribosomes translate secretory and membrane proteins whose genes comprise about 30% of the genome. And we've known for some time that the ER's significance in protein biosynthesis, folding, transport, and degradation. But what was not clear prior to our ability to image with GFP technology was the ER's overall spatio-temporal dynamics. That became very possible with the ability to introduce a GFP tag protein that localizes to the endoplasmic reticulum, or ER. So what you're looking at here is just a simple movie just showing the edge of a fibroblast and the pattern of tubules. This sort of complex tubular array is the endoplasmic reticulum. And you can see as it's extending out to the fine periphery of the cell. Now, when I first started using GFP as a way to sort of probe organelles, basically the ER was one of the first organelles that we actually looked at. And one of the things that I was quite interested in was just how are proteins moving within this organelle. And so in order to address that question, we developed, and this is way back in 1996, we developed a confocal-based approach or strategy for photobleaching discrete populations of molecules in the cell. And so basically here's a cell that's expressing an ER resident protein. We're going to zoom up, we're taking that box that you can see in the center there is going to be bleached. And we're going to watch what happens to the molecules after you bleach. So basically, we use the confocal microscope's ability to shine an intense laser light on a small area of the cell. That led to these molecules suddenly undergoing photobleaching. What was really exciting was to see that fluorescence very quickly returned into that photobleached box, and it returned through a diffusive process. If you look carefully at this movie, you can see that the molecules are moving in from the rim. basically what's going on is that the bleached molecules are diffusing out of the box, and unbleached proteins that are distributed and moving very dynamically within this meshwork of ER are moving back into that box. And so basically, once we saw this phenomenon, we realized that we could use it as a way to measure diffusion coefficients for different proteins. And so this is just a list of different proteins localized in different environments within the cell, and comparing it to how a GFP protein, which is a small, a small protein that doesn't really have elaborate sort of extensions from itself, how it diffuses in water. Basically, what we found is that the GFP, when it's attached to an ER membrane protein, is diffusing at about 0.3 microns squared per second, which is, from model systems, basically what you would predict for a transmembrane protein embedded in a membrane bilayer. And interestingly, that is about 10 to 20 times slower than proteins, GFP proteins that are targeted into the lumen, the inside of this tubular system that comprises the ER network. So basically, stuff inside the ER system is diffusing much faster than the stuff on the membrane. That's not surprising, because membranes are very viscous, molasses-like structures. What was also interesting was that the GFP that was in the ER lumen was diffusing actually slower than GFP in the cytoplasm, but both of them were diffusing significantly slower than GFP in water. So basically, it immediately allows you to start conceptualizing the different types of global environments that molecules are experiencing in these different compartments within cells, whether it be in the cytoplasm, inside the lumen of an organelle, or on the surface of membranes of organelles. Each environment has very distinct effects on the ability of proteins to be able to diffuse. Now, focusing back on the ER membrane, one of the things that we found using a variation of photobleaching, where you continuously photobleach a single area, in this case, a boxed area, we're just continuously photobleaching it over time. And when you look at what happens to areas outside that photobleached box, you see that those areas actually become diminished of fluorescence. And that diminishment or loss of fluorescence in those surrounding areas is due to connectivity of the molecules that are inside versus outside that box. What this experiment told us was that the organelle that we call the endoplasmic reticulum is a continuous structure. It's a single compartment. It's not made of a bunch of different tubes that are sort of separate from each other. It's one discrete structure, because molecules can diffuse throughout that whole structure. In fact, over the course of about 5 to 10 minutes, a single molecule will have explored the entire system throughout that whole cell. So this is really sort of deep information that gives us a perspective of how molecules are experiencing their environment in a particular compartment within the cell. Now, the use of photobleaching is a really valuable way to allow one to look at sort of the dynamics of proteins, because you can remove a population or make them invisible by photobleaching, and you watch how the non-photobleached molecules behave. But an even better way of doing this is using photoactivatable fluorescent proteins. And one of the first of these to be described is the photoactivatable GFP, which was developed by George Patterson in my lab many years ago. Basically, George just mutated all of the residues around the chromophore of GFP wild-type GFP and found a single point mutation that made GFP invisible at 488 nanometer light, which is the normal wavelength that you would image GFP at. But if you excited with UV light, the cell that was expressing this GFP, the chromophore would convert to a state where it was now excitable at 488 nanometer light. So you could suddenly switch on the molecule simply by illuminating with UV light pulse. And this was super exciting because now we could do sort of the inverse of photobleaching and have much more control of when the different discrete populations of molecules that we might want to visualize in cells. Well, why would we want to do that? Well, here's an example. Here's a cell that was expressing a secretory protein, VSV-G, that we tagged with photoactivatable GFP. This protein normally moves through the secretory pathway pretty quickly and distributes out to the cell surface. It's very difficult to isolate discrete populations of this molecule as it's in different stages of its transport. If you're just looking at a steady state population of it being expressed within cells. What this experiment is showing is how we can switch on a discrete population, in this case, the pool of the molecules when they're localized in the Golgi, and now we can track specifically that pool of molecules as they track out and transport intermediates out to the plasma membrane and get dispersed in a sort of a diffuse pattern across the surface of the cell. So this is one application of these photoactivatable GFPs. Another application is to look at protein turnover. Basically, by introducing a photoactivatable fluorescent probe to a protein that turns over that you're interested in studying how fast it is degraded by the cell's degradation machinery, you can precisely measure those protein turnover rates. So in this experiment here, we're looking at the T cell receptors delta subunit that, when it's expressed as an orphan subunit within a cell, will undergo a process called ERAD, ER-associated degradation. And it's known from biochemical studies that the protein will turn over within about 30 minutes. So we wanted to see if we could quantify and look at how this process is working in a more rigorous quantitative way. And the photoactivatable GFP really enabled that. So the first panel in this time lapse that you're looking at is where we UV activated all of the molecules that were in the endoplasmic reticulum. And we then start imaging without any further photoactivation. So we just image, we activate, and then we image. And because there's no new pool of molecules that are being photoactivated, we're only tracking the pool that had been photoactivated, and we can look at the turnover of these molecules. You cannot do this experiment with just a conventional GFP because it's continuously being synthesized at the same rate that it's being degraded, or at some rate relative to its degradation, to create the steady state pool that you will see over long periods of time. But with the photoactivatable, we just switch on the existing pool, and any newly synthesized molecules are invisible because you have not photoactivated them. So here we can actually get thousands of data points in individual cells to look at this degradation rate, and it really revealed a lot of characteristics of this process in terms of showing that it's a logarithmic degradation process, and it's very similar in all of the cell population that we're looking at. Now, around this time, in terms of the sort of history of GFP technological development, in fact, very soon after the photoactivatable GFP really became popular, people began realizing that you can visualize single GFP molecules using, conventional microscopes, simply by isolating, essentially plating the GFP molecules at low enough density that individual molecules are separate from each other. And when you do that, you can realize that you can get very precise centroid fitting of the diffraction-limited blurry spot called the point spread function through this, as I mentioned, through centroid fitting, and get a very precise estimation of where the individual molecule was. And people use this approach to then, in living or in vitro systems, look at how GFP molecules that might be attached to a motor protein are actually, how the motor protein is taking individual nanometric steps, because you could, by fitting the centroid and watching how the centroid is moving in an in vitro system where the GFP may be tagged with a motor, you could get step sizes of molecular motors like dynein. So it was super exciting what the possibilities were using this approach. But the problem came when molecules were too closely associated with each other. And under these conditions, the PSFs overlap, and that prevents them from being distinguished from each other. And I mean, many of you now are just saying, whoa, we know how to deal with this. But basically, at the time, people didn't know how to deal with this. And it was only really with people, including Eric Betzig, who began to realize that this problem could be solved with the photoactivatable GFP. So the idea is to switch on individual photoactivatable GFP molecules, localize their centers, and accumulate the coordinates to create a super resolution image of large numbers of densely packed molecules. And this became very exciting because it allowed you to, if you collected enough single molecules of a structure of interest, it allowed you to begin to do what we call super resolution imaging based on single molecule localization. And that actually led to the Nobel Prize being given to Eric, as well as W.E. Morner and Stefan Held. In the case of Morner and Betzig, it's a single molecule super resolution. For Held, it's a different technology that I'm not going to talk about today instead. But basically, they're all breaking the diffraction limit of light to be able to look at structures with higher resolution. I just want to, as an aside, say that the process of developing PALM was just really a super exciting experience for me. Basically, we were very fortunate to be able to, basically Harold Hess, together with...
priorise pas plus, c'est les demandes fonctionnelles des commerciaux. Et de Sarah, c'est cela qu'il faudra
Excuse me, Stefan?
Oh, sorry.
Right, okay, great.
Oh, good. Anyway, this is just a quick slide to show basically in order to get this technology moving required actually input from a lot of different people. And I think it's important to think about that because I know all of you, at in the Department of Bioengineering are, part of, recognize how many of these new developments are not something that just you can have an idea, but you need lots of people. You need teamwork to get things actually working. And so I'll shout out to Mike Davidson, who was a huge contributor to this whole process by providing photoactivatable probes, originally developed by George Patterson, but also George, who was playing some really critical role with his biological hands in enabling the scope that Harold and Eric had developed, and that's the scope in Harold's living room, to actually be able to do some imaging. So basically, this is some of the first images that we collected using PALM. To the left is a raw image series where individual molecules, in this case, these are lysosomal membrane proteins that are tagged with photoactivatable GFP. They're being switched on one at a time. The center is just showing you the blurry, diffraction limited image that you would see if you didn't have the ability to fit each of those individual photoactivated spots. And to the right is the PALM image that's emerging, the super resolution image that's emerging of the membrane outline of the lysosome. Now, at advanced, basically PALM and as well as STORM and many other variations of single molecule localization microscopy really flowed out from this type of work. One advanced 3D version of PALM is shown here. It's interferometric PALM, and what you're looking at is the endoplasmic reticulum, where we're fitting the membrane shaping protein called reticulon distributed across its membranes. So basically, this is sort of an illustration of how sort of imaging sort of evolved over the course of time, from the perspective of, you start with a GFP molecule, you start modifying the molecule, suddenly new characteristics of these molecules become, real, so to speak, and you can then build on that new technology. So it's basically really, I think, a nice representation of the way science actually progresses, and it's for me a big lesson in terms of moving forward, how you think about where you want to go or where the field is going. So one of the questions that we've been interested in, aside from super resolution imaging, is how can we start looking at many organelles simultaneously? And again, there was a challenge that we have with that. Although there are, a palette of GFP color variants that are available, and this is thanks to Roger Chen and other probe developers. The problem is that the excitation and emission profiles of these probes significantly overlapped. And so that really had a huge, sort of put a big barrier to ourselves and other people being able to do multi-spectral imaging. And so this is just an example where you have 6 different organelles that are tagged with different color variants. We've imaged them with different emission wavelengths. And you can see in the bottom panel of this 6 labeled cell, so basically a single cell is being transfected with all of these different constructs that are expressing different color variants of the fluorescent probe. But what you can see in the bottom panel is that you can't distinguish any of these organelles from each other because of this overlap in the emission wavelengths from these different fluorophores. And so my lab was very interested in trying to overcome this, to be able to really do dynamic imaging. And so postdocs, Alex Valm and Sarah Cohen, basically employed a computational multispectral unmixing approach. This type of unmixing strategy is now available on many confocal microscope systems. But basically, the idea is when you transfect cells in, when you have 6 or more fluorophores that are being expressed within a cell, if you know the spectra of these fluorophores, as shown to the right, you can then use that information to interpret an observed pixel spectrum to precisely identify what subset of the known, expressed fluorescent proteins are giving rise to that pixel spectrum. And in so doing, what you can then do is create a multispectral image of the distribution of all of those distinctly or discretely labeled organelles in live cell movies like this one. So basically here, we've got the 6 different organelles that we've tagged with different fluorescent probes, different color probes. We do our unmixing and we then image, or we image with a lattice light sheet microscope, and from that data set we can unmix these individual organelles from each other to create this multispectral time-lapse movie. Now, this was exciting because people hadn't really seen all these organelles together at one time. You could see them in an EM image, but you didn't see it in light microscopy. You typically see, oh yeah, here's ER, here's mitochondria, but to see all 6 of these organelles at the same time opened up all kinds of interesting questions that could very quickly be quantified from this data set. So these, you got a bunch of different measurements that you can make. Because we're doing this with a lattice light sheet microscope, where we can have close to isotropic 3D information across the depth of the cell, we can come in and quantify things like the numbers of each of these different organelles, because we can segment out all of the organelles from and distinguish them from each other. So we can measure their number, their volume, the speed of movement. You know, for instance, we see lysosomes are moving significantly faster, twice as fast as lipid droplets and peroxisomes. We can see that the endoplasmic reticulum, by looking at the volume of all these organelles, is 30 times the size of the Golgi, 9 times the size of mitochondria within cells. Importantly, we can also map out how these organelles are touching each other or communicating with each other by contact sites, close contacts. And this is illustrated in the top panel series, where you can see each color-coded organelle and its relationship with other organelles. And when we saw these organelles making this type of close contact, if those contacts were being maintained for 9 seconds or more, we essentially define them as a organelle-organelle contact, where basically this presumption is that they're being tethered in some way. And summarizing that data set in the bottom matrix representation, we could define what we call an organelle interactome, which is just measuring the frequency of interactions or contacts that different organelles are making with each other. And so let's look at, in particular, the ER-mitochondria contacts, which are the most predominant within the cell. And to sort of illustrate that, what we've done in this movie sequence here is to segment out mitochondria throughout the entire cell. And now we're going to look at the surface of that mitochondria and when the endoplasmic reticulum makes contact with that surface. And that's what you see to the right in yellow or yellow-green. So all that yellow-green on the surface of the mitochondria represents sites where the ER is making contact with that mitochondria. So that was for me, it was really exciting to see this, because what you can see is that these contact sites are dynamic, but also sustained over fairly long periods of time, consistent with a really fundamental role for communication between the ER and mitochondria. And by communication, we are thinking in terms of lipid trafficking from ER to mitochondria, as well as calcium movement between these 2 organelles, or metabolism, metabolic clock. The 2 organelles are not fusing, however. They can make contact, but they don't fuse, and they don't fuse because they don't share snare components. And this is critical because otherwise you'd have no distinct character. I mean, the organelles wouldn't be able to remain there, maintain their identity. Okay, the other thing that we could do with this type of data set from the lattice light sheet is we could start monitoring the position of the organelle over time. And when we did this for the endoplasmic reticulum, we got a surprise, and that is that the ER at any particular time is occupying about 25% of the cell volume, excluding the nucleus. But what we found, if we tracked the position of the ER and just plotted it as what you're going to be seeing right in this movie, that it explores over 97% of the cytoplasm in about 15 minutes. So there's dynamism associated with the ER, and that dynamism basically allows this organelle to explore the whole cytoplasm. So that led us to start thinking whether we could actually get some better insight into this dynamism of the endoplasmic reticulum. And again, that was the problem that we had, is our spatial-temporal resolution barrier. Is there a way that we can use new technologies for increasing both spatial as well as temporal resolution to look at the endoplasmic reticulum in greater detail? And so to do this, we used a newly developed technique called grazing incidence structural illumination microscopy that was developed by Dong Li in the Betzig lab at Janelia. This GI-SIM combines fast imaging at a near turf plane with a twofold improvement in the XY resolution acquired by structural illumination microscopy. And what that allowed us to do was to really get some spectacular movies seeing the dynamism of the endoplasmic reticulum. And that's what you're seeing here. This is 25 milliseconds per frame. We can go much faster nowadays, but this was good enough for us to see some very interesting characteristics of the ER. We can see that it's undergoing a type of oscillatory motion. That oscillatory motion we found was dependent on ATP as well as GTP levels within cells. We also think it's connected to cytoplasmic forces that might be generated by the actin contractile system. But we think that movement could also be dependent on a very surprising structure, which is intermediate filaments. This is a movie, again, a GI turf sim, a GI sim movie taken by Andy Moore in the lab, where he's looking at vimentin, which is an intermediate filament molecule. And you can see that in the far right panel. These intermediate filaments are distributed throughout the cytoplasm, and their roles in the cytoplasm are really not clearly understood. But what we found by correlating our vimentin images to the endoplasmic reticulum is that there is incredible correlation between these two structures, suggesting that vimentin might be playing an important role in allowing the ER to remodel itself into what we call tight matrices. The center structure where the vimentin knot is, you can see it's just drawing the ER into this tight this tight matrix. All these junctional elements of the ER are just being drawn into these tight areas. We think through some kind of activity mediated by vimentin. Another really interesting set of observations that became possible with GI-SIM was looking at lysosomes, and we can see that they're near ER junctions. In green are lysosomes, the ER is in purple, and microtubules are yellow, and you can see just spectacular organization among these different structures. The ER is being pulled out on microtubules. The lysosomes are clearly adhering to the endoplasmic reticulum, and the lysosomes are actually moving on these microtubules, and we think could be playing an important role in the way that the ER is pulling itself out. So Up to now, I've just focused on how the ER is sort of looking at it through fluorescent technologies that do ensemble, at least in the dynamic sense, ensemble labeling. Two postdocs in the lab, Chris O'Bara and Johnny Nixon-Abel, who was co-mentored by Craig Blackstone at NIH, looked at single particle tracking of proteins in the ER. And I'm going to be speeding up because I think I'm running out of time here, and I still have some things that I want to tell you. Basically, the single molecule tracking became very exciting once we realized that using halo tags to proteins of interest, which could then be recognized by bright photoactivatable dyes, would enable us to track individual molecules, which you're seeing here, in this case, Sec61, an ER membrane protein, but we could simultaneously label the endoplasmic reticulum with a protein that is sort of serving as a serving as a way to visualize the endoplasmic reticulum as a whole. So this is what you can see in terms of these individual molecules, but we can then connect the trajectories, connect the path of these molecules to create trajectories as shown here. This movie's real time, so you can just see how these molecules appear to be diffusing freely throughout the endoplasmic reticulum. Now recently, Chris and Johnny have been looking at proteins that, as I mentioned, could potentially be at these contact sites between the ER and mitochondria. One of these proteins is called VAP-B. It's thought to help mediate the contact between ER and mitochondria. And so when they tagged, the halo tagged VAP-B and started looking at its trajectories within the endoplasmic reticulum, they could see very quickly that the trajectories were not freely diffusing. If you look at the bottom right panel, you can see a single VAP-B trajectory. Part of it is a random motion in the endoplasmic reticulum, but then you'd see hotspots. And those hotspots were revealed to be sites where the endoplasmic reticulum was making contact with mitochondria. And that's illustrated in this movie here, where mitochondria is labeled, a mitochondrial marker is labeled in red. The ER you can see as a whole in white, and the trajectories of VAP-B are being mapped out. And you can see those trajectories undergo very different changes once they reach these contact sites. We can map out the contact sites by single molecule imaging of the trajectories, and that's illustrated here, where we have VAP-B normalized localization probabilities over a 60-second time window. And to the left is just showing mitochondria and its position over that 60 seconds, and to the right is the distribution, the localizations of VAP-B during that time frame. You can see hotspots where the VAP-B is being essentially showing association with these contact sites. So the VAP-B trajectory hotspots, since we can map them out at single molecule resolution, we can essentially map that onto contact sites that you can see by electron microscopy. And basically, as you can see at the very the bottom two panels, the contact site, as seen by single molecule tracking, is very similar to the contact sites that you see by electron microscopy. So in the final part of my talk, I want to turn to electron microscopy, because basically that's the ultimate resolution. There's limitations in terms of dynamics in that system, but you know, if you're interested in the way cells are organized, you got to go down to the, you know, the finest level. And recently we've been collaborating with Harold Hess's lab to do that using focused ion beam electron, focused ion beam scanning electron microscopy. or FIB-SEM. And basically this approach involves cycles of imaging using scanning EM and sample milling by a focused ion beam. And this allows cell visualization at up to 4 nanometer voxels, which is small enough to start analyzing organelles like the ER's 3D complexity at isotropic resolution. An example of this is shown in this on the right-hand panel here, where we segmented out the ER in a FIB-SEM volume, roughly the size of a confocal volume. And the confocal volume equivalent is shown in the green at the bottom, where basically everything in that confocal volume is flattened. But you can see that the FIB-SEM in that which shows the 4 nanometer distribution of the ER within that confocal volume reveals a very different type of ER than what you would see by a confocal. Confocal looks like it's a flat structure, branch structure. In fact, from FIB-SEM, we can see it's a very complex, elaborate, three-dimensional structure. So given that recognition, at Janelia, we've been working with a team of people to try to make sense of whole cell FIB-SEM data sets. And this is an example of a whole cell EM volume that's being acquired with isotropic resolution, because we're the resolution of the scanning EM is equivalent to the sections that we're collecting, which is about 4 nanometers. When we do that, we can come in and start looking through these sections, and we start seeing incredible diversity of these different organelles. What you're going to be seeing in this movie is manual segmentation of all of the different organelles that you're seeing isolated to the left. So you can see in this, you can see the Golgi apparatus in red, the endoplasmic reticulum in green, the multivesicles, lysosomes in purple, and the nucleus at the very bottom. Now, this particular volume set took around 3 to 5 days to actually segment by a single person. If we want to do a whole cell, which is 7,000 times the size of this volume, we estimate a single person, it would take them 50 years. So that's just not going to happen if you really want to be realistic in trying to understand how all these organelles are distributed at this resolution within a cell. And so as a consequence of that, we've at Janelia begun generating a team that can do whole cell organelle analysis with the goal of classifying and segmenting all intracellular organelles. And so to do this, you have people who segment, we have people who are, and the segmentation is to provide ground truth to how you annotate particular organelles. And then we have a modeling team that comes in to use deep learning algorithms to try to see if we can do this automatically. So segmentation team first manually classifies, annotates, segments all the organelles in several regions of the cell, and then the modeling team uses this as ground truth to develop machine learning. So here are some of the manually segmented regions of the cell that were used for training the deep learning architectures that enabled automatic segmentation of the 13 structures of the cell shown to the left. And here's the predicted segmentations and automatic reconstructions throughout an entire cell are shown here for 5 different structures, which are plasma membrane, ER, mitochondria, endosomes, and nucleus. As you can see, the organelles are well predicted by the machine learning algorithms. And from these data sets, we now have the ability to extract quantitative information, including the instance count, the volume, surface area of each of these organelles. Now, I just want to emphasize just how powerful this is in the next 2 minutes, if you can give me that. Here's an example where we're looking at one particular subcompartment within the cell, which is the sites where the endoplasmic reticulum releases its cargo, and this is ER exit sites. And this work has been done by Aubrey Beigel and Qilin Chang in the lab. Aubrey is head of the COSIM team and Qilin Chang is a postdoc. Basically, they could, I'm sorry, let's just go back here. They could, by doing cryo structural illumination microscopy to identify ER exit sites, which are the purple structures, they could then come in and segment out at very high resolution the organization of the membranes at these ER exit sites. And what they found was that the ER exit sites seem to be comprised of a highly entwined tubular network. The microtubules were often seen in association with these sites, and this was characteristics of hundreds of ER exit sites. So every ER exit site that we could see within the cell had this type of characteristic where the average diameter of the exit site was around 360 nanometers, and the tubule diameters were about 40 to 60 diameters within that structure. And the tubules connected to the rest of the ER, which is blue in this image, through single necks. We could also see that these ER exit sites enlarged upon cargo release. What you're looking at in these two different images is 8 minutes after release of a bolus of cargo from the ER versus what it looked like before. Big change, again, underscoring the dynamic characteristics of these structures. This is just showing how we see that the tubule diameter as well as the overall size of that exit site can expand. We can also use FIB-SEM correlation with probes to look at transport intermediates that are moving from ER to Golgi. Here is a fluorescent probe that we introduced that can be released from the ER through a Bioengineering, and 8 minutes after that release, you can see all these tubules pouring out of these ER exit sites. If we then sectioned through the cell and identify where those fluorescent tubules are, we can then segment the areas. We can then visualize these tubules at this nanometric level by FIB-SEM. And that's what you're seeing here for 10 different structures within the cell that we just picked. Here's one of these structures. That's the overlay of the cryo, the structural illumination image, and here is the characterization of that structure. And these transport intermediates, I think what was interesting about them is they're almost always associated with microtubules. You could see that in the FIB-SEM data set. And they're also connected oftentimes to the ER exit site itself. The same characteristics could be seen for endogenous transport intermediates, where we're not artificially introducing a fluorescent probe. We can actually see these things just in any kind of, in the cell that has not been overloaded with cargo. So basically, this is the model that we were able to come up with for ER exit site ultrastructural organization based on this FIB-SEM reconstruction of the cell. So to sum up, I just want to say that imaging is just a continually evolving field that can explode our different parts of the cell. There's tons more to do in this world. I think it's important to be, if you are just starting out in this field, what I would say is take, think hard about questions, because the questions frequently can drive the need for new developments, but also, pay attention to new developments and see whether some of them can address particular biological questions. And so that only happens when you have collaborations between the biologists, the engineers, the chemists, and that basically has been one of my philosophies throughout my career is to have an ongoing conversation with different fields to see how you can attack basic fundamental biological questions using the incredible resources that we have now available to us. And with that, I want to thank you for your attention. These are people who participated in the different technologies that I talked about. I'll just shout out my single particle tracking people, Chris Obera, Johnny Nixon Abel, the TURF-SEM imaging, multi-spectral work, Eric Betsick, Sarah Cohen, Alex Valm, and the FIB-SEM work, which is now really being pushed by many people at Janelia, including Harold Hess. Thank you very much.
Thank you very much for this, Jennifer. It was really awesome, actually, seeing all these techniques and everything. It was amazing. Thank you very much. And now we can go probably to the Q&A session. We have some questions in the chat, but feel free if you have questions to raise your hand in Teams, and then we can be there and ask them. Should I start first with the ones that we have in the chat? Could be an option. So Chris Rollins asks, one of the primary limitations on PALM storm resolution is increasingly the expression level of the probe. It's hard to determine whether a membrane is continuous without the very high labeling density, for example. Are there any potential strategies to overcome this problem?
Well, one is to do correlation with electron microscopy. So, you know, one of the things that you can do, I didn't really talk about this. I was talking about doing cryo FIB-SEM, where you take an image, the way we do these FIB-SEM experiments is the cells are high pressure frozen before we do our sectioning and scanning electron microscopy reconstruction. But you can introduce a step before the electron microscopy step, which is to image these cells using conventional confocal microscope with a structural illumination microscope or PALM or STORM. You have to do that at cryo temperatures. That's the key thing. But it turns out that many of these fluorophores are very bright and stable at cryo temperatures. So you can get spectacular PALM images if you're imaging a cryo fixed sample. And so this is where I think you can overcome, you know, this type of problem, because then you just take your PALM or STORM image that you collected at the cryo temperature and superimpose it on top of a FIB-SEM reconstruction of that region, and then you get all of the framework. You get all the membranes that you want to look at EM resolution. So that's the strategy that I would take. I mean, another possibility is to come in. You could do possibly double labeling with a lipid dye. If you don't want to do electron microscopy, you could get many thousands more molecules with a lipid dye that would give you sort of a brief outline of a membrane, and then you superimpose your protein distribution on top of that. I mean, that's another possibility, sort of using a type of paint. You could paint the membranes with a lipid probe that can bind and dissociate from membranes, and when it's bound, it fluoresces, and you can fit it. This is a technique that Eric developed a while back, where he used it in combination with a lattice light sheet to do 3D mapping of membranes within cells. But you could then do PALM on top of that to look at particular protein populations. I hope that helped the question, answering the question.
Chris, I don't know if you want to ask something. Maybe you can unmute yourself. It's you, Chris, as well.
No, just a fantastic answer. Thank you very much.
Okay, cool. So the next question is from Lucky, and he asks about the spectral mixing. How was the spectral mixing accomplished? Typical approaches using linear mixing or principal component analysis are computationally challenging, and their performance degrades as light levels decrease.
Yeah, so I think that the concern is that, and this is a concern, if you are if you're trying to unmix 6 or more different fluorophores that you've introduced, if any of those fluorophores are super bright or super dim, they can completely mix, they can overwhelm your principal component unmixing strategy. So it's absolutely critical in doing these experiments that when you've got to do a lot of troubleshooting in terms of knowing exactly how bright to make each. You have to express the proteins at relatively similar levels so that their fluorescence doesn't dominate when you do this unmixing. So that's the problem that you face with this approach.
The other question is again from Lucky, and maybe Lucky, if you want to unmute yourself and ask the second one, or then I can read it. I don't know, because I think, okay.
Hi, Jennifer. So I have another question is about the organelle dynamics you showed. Have you considered the application of, for the question about the organelle dynamics, have you considered the application of optogenetic techniques to investigate, let's say, how the organelle network responds to altered behavior of one or more of these chosen organelles?
No, that's cool. Great idea. Yeah. Yeah, so if you suddenly drag an organelle over to one side of the cell, how do the other organelles respond? We haven't done that. It'd be great to look at that. I could make some guesses as to what I think might be going on, but I mean, I think it'd just be fascinating. These organelles are all, well, actually, I could say one thing. We've done one experiment that could be sort of related to what you just said. We were interested in swelling cells as a way to swell organelles to make them big and round. And you can do that by removing water. Basically, I'm sorry, adding water. So you basically take your cell and put them in, dilute your medium tenfold. And what will happen is water rushes into the cell and the cells swell. The cell swells, but the organelles swell. And what we found was that organelles that were tethered to each other, that were making contacts with each other, stayed together. They didn't blow apart as the cell expanded. And so we could look at these contact sites between different organelles, and we could show that actually those contact sites have a very special lipid environment associated with them. And we could show that certain proteins were associated at those contact sites. So that, I think, is an example where we have done a perturbation. It's like a sledgehammer, but it enabled us to see how these it was surprising, the organelles stayed together. So these tethers are strong enough to keep particular organelles it keeps them together even though everybody else is being blown apart or swelled away from each other through this osmotic change. Interestingly, if we take the cells back to normal medium, everything comes back. The cell actually can reform. Basically, the endoplasmic reticulum sort of refuses, rearranges itself. But if the mitochondria has herniated, the cell dies very quickly. So it's a matter of, the cell won't die when it's swollen. When you bring it back from being swollen, the mitochondria, if it has herniated, will kill the cell through apoptosis very quickly. Anyway, it's sort of interesting.
And the next one is from Emmanuel. have you considered expansion microscopy to study the ER? Out of curiosity.
Yeah. Let's see. We haven't. I mean, we played around with that, but we haven't done it in a serious way. But I mean, it's a possibility. Absolutely. I mean, what I think is important is each of these different technologies, including expansion microscopy, has particular sets of questions that it can address and other questions it can't address. And so with expansion microscopy, that's where, for me, it's like, okay, well, what's the question that I really want to address here, that I can't get in a much better place. And since we can do electron microscopy and see the ER in its natural context with, high pressure freezing, there's nothing that expansion microscopy is going to tell us more, except maybe something about the way proteins are distributed. So, if you can get some really interesting protein labeling patterns, I mean, that's the one hiccup with the electron microscopy. It's hard to see distributions of individual proteins in the way that you could with expansion microscopy, or PALM or STORM.
And the next one is from Naomi, but maybe Naomi you can unmute yourself and ask if you want live.
Hi, it was beautiful talk. Thank you very much. So my question is kind of general, but as you so nicely walked us through with the evolution of the imaging technology, the resolution gets better. And did you see any changes in the measurement of the same thing, like GFP diffusion in the cytoplasm versus ER membrane lumen? Is it always the same, or with the better resolution, did you see sort of a change?
Well, we found, I mean, that was very, really interested in that question. Basically, they should be the same in terms of the diffusion coefficient, or else. But I think you have to realize that when we do the FRAP photobleaching, A, it's an ensemble measurement. B, our diffusion coefficient is actually making assumptions about the structure of the ER. And in the paper where we made that 0.3 micron square per second claim for diffusion coefficient, we were assuming the ER had a particular structure, a reticular structure, but that reticular structure was uniform throughout the whole system in terms of the way we were analyzing our ensemble recovery rate.
Yeah.
With a single molecule, you're seeing the whole thing. So it's a different. It's like it's comparing apples and oranges.
Yeah. Okay. Thank you.
So it's different questions. Like, I'll tell you something really interesting with a single molecule. So, the ER is this tube, and we found that certain proteins like to move like this more than this. So some proteins want to move, they want to slide along in sort of a lateral direction.
Yeah.
They'll go randomly, but they'll just go this way. Other molecules want to diffuse this way.
Yeah.
And a lot has to depend on the topology of the protein and the way it's integrated in the membrane. Yeah. So that was really interesting. And what we found was that the proteins that like to go around in circles, those guys in a FRAP experiment look like they're moving much slower because they don't get very far.
Yeah.
If you treat the ER as one big, two-dimensional plane.
Yeah.
But they, in fact, are moving as fast as the other molecules. But they just prefer moving in a particular topological domain on the surface of that ER. So that was an interesting realization.
Yeah.
And so that's a perfect example of where we assumed that protein was really slow based on FRAP, but in fact, it's as fast as the other molecules. It just prefers moving on a different surface topology of that organelle.
Great answer. Thank you so much.
Yeah.
So we have also Dane Wolf has an equation. Maybe you can unmute yourself and then you can ask directly. And sorry for not giving you the asking actually earlier for this one because you raised your hand. Can you unmute yourself, Dane Wolf? Dane?
Says trying, but maybe another person first, maybe.
Yep. So then maybe Jacques and Lee, do you want to answer? Maybe you can unmute yourself and then ask them.
Yes. Thank you so much for the seminar, and I'm really inspired by your talk. I have a question regarding a small detail for your paper analyzing the ER dynamics with high resolution. So I noticed in the figure, you show that the ER tubule somehow has a bulk movement, that the ER luminal proteins are not evenly distributed even in the same tubule. And then I'm wondering why the cell need to arrange this ER tubule this way, and what are the biological function meanings for this? And then in terms of, because this special constriction, because you have shown that reticulin shape the ER, but do you know how this reticulin can somehow find a very small spot on the ER tubule and shape it further, but not other spaces? Thank you.
Yeah. Well, as I mentioned to Naomi, some of these proteins, reticulons in particular, are hairpin, and they prefer like going around in circles. They like high curvature, so they're spending a lot of time moving along. If this is, here's the tube, they like to go like this on the tube more than like this. So that can lead to them actually ending up in places where there's a lot of curvature. And so that's the only explanation that I can give at this point. But your other first question is, to what extent does these different morphologies of the ER play functional roles in terms of the ER? And that's something that we're very interested in right now. We have some work that we're currently doing where we're looking at mRNA that associates with ribosomes on the surface of the ER. And we're seeing that the mRNA is moving very differently depending on where it's localized on the surface of the ER. And in particular areas of the ER, the mRNA is preferentially translating, where it's gotten enough information to start translating a protein through the ribosome. So that's where we're headed right now, is trying to relate the structure of the ER to some of these really fundamental roles of the ER in translation of proteins.
Thank you so much. Can I ask a very short follow-up question, if this is possible?
Yeah.
So the follow-up is about this special arrangement of reticulin at the foci, where it's, it's the moving. Because in the figure, you show that where the ER is more constricted, is then be released within milliseconds. So at one side, you have constriction and release constriction within a few milliseconds. So if reticulin is executed this constriction for a milliseconds, do you also see the dynamics of reticulin having this spiral movement within few milliseconds?
We haven't looked at that.
Okay.
Good question.
Okay, thank you so much.
Yeah.
And Dane, but now probably you can unmute yourself, right?
Hey, can you hear me? Sorry about that.
Yeah.
Dr. Lippincott-Schwartz. I don't know if you remember me. We met in Kyoto in 2018. Anyway, I really enjoyed your talk. I'm particularly interested in how you showed the ER is very dynamic. And as we all know, the ER has a close association with mitochondria. I'm wondering, do you know whether the dynamics of the ER, insofar as they are ATP dependent are driven by associations with mitochondria? In other words, could there be like an ATP gradient coming from mitochondria, which is driving ER dynamics? And then could you actually determine whether ER dynamics close to mitochondria might be more dynamic, or there may be more movement or more fission fusion near the ATP source, as it were?
Yeah. Yeah, I mean, that's a really good question. We haven't really addressed that. I mean, certainly there's some very beautiful work that Jody Nuneri and Gil Volz have done in terms of the relationship of ER and mitochondrial division. The thinking is that connects to actin recruitment and dynamine-like protein recruitment to these sites. So I'm not sure exactly. I mean, certainly the ER dynamics are going to be critical for the ultimate, sort of ability of the mitochondria to pull itself apart at some level, potentially through orchestration of these cytoskeletal components. But yeah, we haven't taken it to any further level.
You don't know if the inverse might be true. I mean, for an ER tubule to really move quickly, does it need to be close to a mitochondrion, or for an ER to undergo fission, is it more likely that there will be a mitochondrion nearby which could be supplying the energy for those processes?
Yeah, I don't think so, because yeah, I'm not, the energy, mitochondria is not the only place where you make ATP within the cell. And there's glycolysis that's going on all over. So you need to know where the ATP is. You can, I mean, you could shut down glycolysis and then look to try to address your question. But we'd have to shut down glycolysis with deoxyglucose, for instance, and then look and see whether there's some correlation between mitochondria and what the ER is doing, or vice versa. But I think you have to realize that there's two different ways cells get their energy, and that's something that I think is really interesting. And yeah, I mean, basically, if you really want to understand cells, you got to understand the energy flow through this system, and because basically everything we see in the cell is a consequence of the way energy is being handled, flowed through the system. So it's a good question that you're asking.
Is it known? In other words, is glycolytic ATP versus mitochondrial ATP the main driver of ER dynamics?
Probably not, but it could be the main driver of mitochondrial dynamics. Because when we starve cells of glucose, we see mitochondria tubulate, mitochondria expand under those conditions. And mitochondria are still doing energy production under those conditions. So yeah, no, I mean, it's a good question. How are these different organelles coordinating with each other depending on the energy source that's available to the cell?
Thank you
very much.
Yeah.
And one last question that we have here. Ravi Shankar, can you unmute yourself and ask the question?
Thanks a lot. Thanks a lot for the wonderful talk, Professor Jennifer. I'm from India. Well, it's been fabulous to listen to the talk and what COVID gave an opportunity to the Zoom calls and all. So great that you showed the evolution from the photoactivable GFP all the way to development of various techniques and all that. I have one question with respect to that. Another one about the ER dynamics. The question about the techniques that you're talking about is, can you use that to to measure large scale the turnover of various proteins? Okay, that's one question. The other one is about you mentioned that the ER has a very fast diffusion rate. It covers like within 15 minutes, it covers the entire cytoplasm. So are there any preferential turnovers or preferential dynamics that's happening when it's in different locations in cytoplasm? Is there something that you have looked at?
Yeah, no, that's a good question. The only thing that we can think of that we're not really pursuing it, but the most obvious thing would be, the ER is a big calcium sink within cells, and it's also sensing calcium. Calcium is a very important signaling molecule within cells, and the ER can take up calcium through creating contact sites with the plasma membrane, because there's lots more calcium outside the cell than inside the cell. Basically, you want to keep calcium really low in the cytoplasm because there's phosphate, and phosphate is critical for making ATP. If you have calcium, phosphate precipitates out. So calcium has to be really kept low. And so the ER is sopping up the calcium all over the place and potentially distributing it to different organelles, because mitochondria uses calcium in terms of its electron transport mechanisms. The lysosome uses calcium in a lot of enzymes. So the lumen of all these organelles uses calcium to drive various processes. Whereas calcium out in the cytoplasm is it's got to be kept low. And that's one reason why calcium is actually used as a signaling molecule. So I think having the ER move around maximizes the ability for it to be able to sop up calcium or release or distribute calcium to different places within the cell. That's my gut feeling. Your first question I didn't understand. Maybe you could just repeat it.
Yeah, so it is about using the techniques to measure turnover rates of proteins overall.
Okay. Yeah.
Okay, like a whole lot of proteins or
yeah.
at a cell level.
Yeah, that was just the strategy is with the photoactivatable GFP. You just express it on a protein that you're interested in looking at its turnover rate. You then photoactivate it, and you just watch the fate of the photoactivatable pool. It's a powerful way to look at turnover. Is all I was saying in the talk.
All right. Has anybody done it in like a scaled up manner, so that because the turnover rates of many proteins are kind of still not known, right? So
yeah, no, nobody has. No.
It sounded like an exciting technique to be doing that. Actually, people use mass spec and other kind of approaches to be intensive.
Yep.
Okay. The third one would be a comment. Very exciting to look at the kind of work that you're doing with the AI machine learning, applying that on the electron microscopy data. Well, I'll ping you up if there is any possibility to collaborate, especially in terms of annotations and all. We do look at a lot of brain imaging data sets, and this may be some interesting data to look at, large scale data.
Yes, this is a very, very exciting. And I should say that Janelia is, we're very collaborative in that respect. And especially moving forward where there's a new research area that is emerging at Janelia, where we're going to be focused on looking at cells outside of tissue culture, looking at cells in the context of living organs, organ systems, tissues, inside whole animals, and using these technologies to see how cells contribute to the physiology of a whole system. So that's a new direction for Janelia, and there's going to be lots of collaborative potential there. So yeah, please keep us posted with your interests.
Great, thanks a lot, Professor Jennifer, and thank you for the opportunity.
Yeah. Oh, Constantine, I think you're muted.
You're muted, yes.
Sorry.
Yeah, you're right.
Yes.
Sorry for this. Yeah, and I'm saying that actually I think that we are done with questions right now. I don't see that we have something else in the chat or someone else with something. I want to thank you once more for this great talk, and I hope personally at least that we could see you coming here probably in the future when everything is better.
I'd love it. I always love coming to England. Absolutely, to London, yeah, in particular. Good. Okay, fantastic.
I don't know if you want to say something.
No, it was thank you so much for so you, as you noticed, that our Q&A session kind of become a mingling session at the end. And
yeah, no, that was really fun.
But maybe there are some people who are staying on. They might be wanting to talk with you a little bit more, possibly, if you can stay on. But I'm gonna stop recording this session. Yes, and then yes, great.